Hello, everyone. We're pleased to have you join us for Cardinal Health Lab Exchange. Welcome to today's live broadcast, Assessing Quality Using ACCURUN Controls for Quantitative Viral Load Assays. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's webinar. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by SarahCare. SarahCare partners with IVD researchers and manufacturers, as well as clinical laboratories, to bridge the gap between today's diagnostic solutions and tomorrow's engineering technologies for molecular diagnostics, next-generation sequencing, and companion diagnostics. For more information, please visit www.sarahcare.com. We have a few important announcements before we begin. This presentation has been approved for continuing education credits. Please click on the CE button at the bottom left corner of the auditorium. This will direct you to the necessary site and form needed to receive your credits. This webcast is also designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many of your questions as we can today. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button, lower left. Following the presentation, please make sure to head over to the SarahCare virtual booth here at Cardinal Health Lab Exchange to have the opportunity to engage in live chat and have more of your questions answered on the spot about assessing quality using ACCURUN controls for quantitative viral load assays. I would now like to introduce today's speaker, Russell Garlick. Dr. Garlick is a scientific and R&D leader with over 30 years of experience in life science industry. He has led the development of highly technical R&D projects for new products launched into the research, translational research, and diagnostic markets. He has working experience with plasma, cellular, and cell-based assays using receptor binding, immunoassay, mass spectrometry, and molecular diagnostic technologies. At SarahCare Life Sciences, Dr. Garlick is currently leading the new technology development for IVD controls, as well as reference materials for next-gen sequencing applications in oncology, infectious disease, non-invasive prenatal health, and genetic testing. When he was at Protein Forest, he led the development of an isoelectric focusing chip for protein and peptide mass spec sample prep, instrument and data analysis software. Previously, at NEN DuPont Medical Products, Dr. Garlick led the efforts to develop multiple diagnostic tests used to manage AIDS diagnostics, including an HIV antigen assay and an early drug resistance assay. Dr. Garlick has been a member of AACC since 1992, is an affiliate member of AACR, and is an affiliate scientist with ACMG. He has published 13 peer-reviewed papers and has 2 patents and 2 patent applications. I will now turn it over to Dr. Garlick for his presentation.
Well, good day, everybody. Thank you, Brenda, for that introduction. Welcome to the SarahCare Life Sciences seminar. Today we're going to speak about assessing quality using the ACCURUN controls for quantitative viral load assays. So I'm Russell Garlick. I've been with SarahCare a number of years here. I'm actually located in Milford, Massachusetts. We actually have 2 sites, one here in Massachusetts and one in Gaithersburg, Maryland. So let me go to the learner objectives. Twofold. One, to apply virology controls using sigma metrics for quantitative viral load assays to an overall laboratory quality control system. And I want to bring this to life by using current data that we've generated in our own laboratory, by reviewing some of the virology around HIV-1, HBV, and HCV, qPCR assays, and using controls as part of a laboratory QC strategy program. The agenda will be a few minutes introducing SeraCare Life Sciences, spend a little bit of time talking about the viruses that we're talking about detecting, and of course the importance of quality control, and especially with these quantitative virology assays and uniqueness about quantitative measurements and metrics. We'll talk about using sigma metrics as a way to analyze your data, some specific examples from our own laboratory, and then when assays do fall out, some troubleshooting ideas, and we have a list of references. And of course we can give you this entire presentation through accessing SeraCare. And there's a number of references that you can get access to. So let's move on to SeraCare. SeraCare has been in business for over 30 years supporting advanced diagnostics. Company started back in the 1980s during the AIDS crisis using serial conversion panels to help IVD manufacturers develop highly sensitive and specific assays for HIV diagnostics. The company moved into other forms of diagnostics, including process materials, other controls that would use to validate and develop assays, and then moved as technology platforms changed into qPCR methods, multi-marker mixes for serology controls in the year 2000s. And the company's gone through a number of iterations and a number of acquisitions. Most recently, we're expanding into precision medicine, although I won't speak to it today in today's presentation. We do have a molecular center of excellence in Gaithersburg, and we are focusing on next-gen sequencing applied to oncology, and again, making controls, standards, and reference materials for oncology applications, as well as non-invasive prenatal screening, maternal fetal medicine. So stay tuned for additional products and technology coming from SeraCare in those application areas. So let's spend a little bit of time talking about the viruses. Here's a diagram of HIV-1, which is a retrovirus, as everyone knows. The figure shows the virions, and of course the RNA is a dark spot inside the virions. Now the importance of measuring viral load is shown here, that initial upon diagnosis you want to confirm the HIV viral load to see how long the infection's been going on. And typically in the US, there's another retest of viral load every 6 months. So fairly frequent test frequency for HIV. As well as under therapy. Now under therapy, you can often see the viral loads dropping to less than 50 copies per mil. Now what's really important for HIV is, of course, drug resistance. Over time, the virus becomes resistant to a number of ART therapies, and when you see copies per mil start to increase, specifically above 1,000 copies per mil, then you have to consider resistance testing. And within the literature, external quality assessments have set the lab standard at plus or minus 0.3 log copies per mil. So that's kind of the target we want to be setting for our assays to be able to detect this virus. Now the virus is still a major worldwide health crisis. There are 35 million people living with HIV and AIDS around the world, and still to this day 1.5 million fatalities per year. And even in the United States, numbers of new cases continues to grow. And as you can see, in the year 2011, 44,000 cases of new cases of infection, and most recently 2013, that numbers are actually risen up to 47,000 in the year 2013. So that's HIV. The second virus we'll be talking about today is hepatitis B virus, which is double-stranded DNA virus. Now in this case, the NAT test, nucleic acid test, is an indication that virus is replicating and the person is highly contagious. And typically the treatment regimens, if the HBV DNA is above 2,000 international units per mil, on patients who are hepatitis B E antigen, so this is the core antigen that is actually expressed into the plasma of the patients. If it's negative, then the physicians and patient management, you should start treating for this infection. Now, very large numbers here. Worldwide, 2 billion infections around the world with 400 million carriers. And as you can see by the global map, the red indicates high prevalence rates and green lower prevalence rates. But even in the U.S., there still are a number of acute cases per year, and 3,050 in the year 2013. Now, good news that in the U.S. at least, this has declined from 8,000 new cases of acute cases in the year 2000. So that's hepatitis B. Now moving on to hepatitis C. Now on this image, one thing I wanted to point out, oftentimes you see hepatitis C with its pointy edges on its envelope proteins, and if you look at that virion right in the middle, you can actually see the pointed edges to the virion. Hepatitis C is now an RNA virus and still can be detected with qPCR methods. And typically assays are done for seropositive persons to confirm active infection. Most importantly for drug therapy choices to determine which genotype a person's infected with, and also to follow and monitor treatment. Now in the US, it's 2,138 active HIV infections in 2013, but as we know, the chronic infection rate is very, very high in the United States, particularly in the baby boomer generation, as 65 per 100,000. And over time, these will lead to severe liver disease. But the good news on HCV, of course, is now we have therapies that can actually eradicate and cure the disease. So that's a little bit about the viruses we're going to be measuring. Now let's think about it from the laboratory perspective. Now laboratories are under a lot of pressure. You are CLIA certified, and you're running probably both laboratory developed tests and FDA approved tests. You're always under pressure to add more new assays, which is good because we want to be able to support patients and diagnostics as best we can. So you're under pressures to add assays, but at the same time, you're under pressure to reduce costs. And you want to be doing all of these very sophisticated assays, HIV viral load, HCV, HBV, and all these other assays that you're being asked to deliver. Now, how I like to think about this is, as shown here in the word cloud, there's really two central components of any laboratory's QC program, and that's to keep patient front and center and keep your quality control front and center and up to date. So good quality control will lead to the proper patient results and proper patient diagnostics. So with all of this activity go on, and as we'll talk about in today's seminar, keep QC front and center, and you'll be satisfied with your patient results. So why quality controls? The first reason, there's really three things you want to be able to differentiate. One is to differentiate normal changes from biological variation. And second, we'll talk about treatment. You want to be able to follow a specific patient to make sure they're responding to treatment and viral load is dropping. And from a QC perspective, you want to be able to early identify any assay drift or any assay variation over time. Now, in example, HIV normal variation can be as high as 0.2 to 0.3 logs over time, and that's just normal variation. And antiviral treatment, as I mentioned earlier, is very, very effective, and you can take viral loads actually below the detection limit of the assays. Also, we want to be able to detect small shifts over time, and these oftentimes happen with reagent lot changes, and clinical laboratories have been dealing with these issues for many, many years. Another reason for quality controls is you're required to perform them in a way that helps you reduce your costs without compromising quality. And how you do that is really emphasizing in well thought out lab QC and lab QC metrics. So when assays are within quality standards, it saves time and money and reduces risk. And of course, complies with any clear regulations that we have to follow. So the characteristics of viral quality control materials. And I'll speak to this as a manufacturer. As I said earlier, SeraCare has been manufacturing these types of products for over 30 years. And our strategy to develop these products is to use intact virus that would represent the virus that is being detected in the patient sample. And by knowing the genotype, it actually provides additional information that you need for drug therapy. We also want to make sure that the control is commutable, same patient matrix as the sample. And this allows you to monitor all aspects of sample testing, from extraction to detection to quantitation. And we also pay a lot of attention to the fact that we want you to be able to achieve the right levels. So that means that how we manufacture these products, we want to make sure they go through the proper extraction and method performance over the range of interest of your assays and within clinically relevant ranges. And viral quality control materials, under best circumstances, should be different from kit calibrators to ensure that the QC procedures provide an independent assessment. And I'll speak to this in a bit here. And typically, when you set your assays up or using an approved kit, you always have 2 levels for positive control and 1 level for one negative control. And what we do at SeraCare is we make large, stable supplies, so it allows you to test your assay this week, next month, and in some cases even next year. It gives you a nice reference material that you can use over a long period of time. And as shown here in the lower right, this is right from ISO 15189 requirements. So control materials, so independent QC materials, that's what we produce here at SeraCare. The manufacturer, in this case SeraCare, we set the design of the control based on inputs from what the patient diagnostics needs and the laboratory need. And we view them, and as the industry does, as true external independent quality controls. And our controls have both IVD, US, and CE marks. IVD manufacturers also supply QC material, and let me state here that we do not recommend replacing in-kit controls with our third-party controls. You should always run the in-kit controls as recommended by the manufacturer as well. So the IVD manufacturer makes in-kit controls. Generally, they're designed to work as a system by the manufacturer of the instrument, of course, in the assay design. And they're made in-house to internal specifications and usually have IVD CE marks as well. Now, our brand of NetPositive Controls, nucleic acid testing positive controls, are the ACCURUN brand. And these are manufactured from intact virus. In the 3 examples we're talking about today, HBV is made from DNA positive serum or plasma, and we dilute it to the proper concentration for the specifications. HCV, RNA positive, is also from human or serum positive plasma, and again diluted in a commutable matrix to the appropriate levels. HIV is a cultured virus. It's type B, and it's the very famous virus 8E5 that we actually grow and culture, but again tied it to the proper concentrations in the commutable matrix. And with these NetPositive controls, we provide multiple levels that allow you to challenge the dynamic range of your assays. And in all cases, they're diluted in a patient-like material, making it commutable. And we go through a manufacturing process where we defibrinate, dialyze, and filter our plasma that goes into making our products. And we make sure that they are non-reactive for other infectious organisms. And in all cases, they're release tested on an FDA-approved qPCR assay and available to customers around the world. Now, this diagram helps you as a laboratory to decide how frequently to run a quality control system. So there's 2 options. Either if you're running an LDT assay or an FDA-approved assay. On the left-hand side is the LDT. Or, in fact, if you've modified an FDA-approved test. The standards from ISO 15189 suggest running a daily control to confirm results of meeting the expected results, and run additional controls whenever there's reagent changes or preventive maintenance or replacement of any critical parts, or in fact, if you have new technicians working in the laboratory, or any other changes that the laboratory would see fit to run additional controls. And again, the beauty there is you can compare the results lot to lot with our third-party controls. So those are LDT recommendations, and on the right-hand side is the FDA-approved tests. And with the FDA-approved tests, as I said earlier, you should run the controls as recommended by the manufacturer, and the ISO regulations suggesting running the third-party controls at each new lot or shipment. I would emphasize that in all of the statistical measurements, more data is better. So we really do encourage laboratory directors to run and increase the frequency, even with the FDA-approved test, so you have data so you can do real statistical analysis of trends over time. So once you've decided how frequently to run your controls, then you have to think about how to analyze them. And we make a recommendation, as do other QC experts, to use sigma metrics. And the advantage of sigma metrics, it's a quantitative measure for the quality of a test. It's really easy to monitor, and not only can you follow trends, but it gives you rules that you can follow and establish before really taking your assay live with patient samples. And it's been used in many different areas, from clinical chemistries to serology, and it's a very straightforward approach. It's very well proven in many types of laboratories. And ultimately, like we said earlier, the goal of any QC program is better patient care, and with less failures, reduction in cost, and reduction in risk. So the sigma metrics roadmap, it's pretty straightforward. You need to collect your test characteristics. You want to collect and calculate a sigma value, and this is taken from other industries. A sigma value of 2 is not a very good assay, and a sigma value of 6, which is super world class. So in most cases in a clinical laboratory, the assays fall in the range of 3, 4, in some cases 5. But once you've established your metrics, then you select an optimal rule. It helps you guide decisions with your assays, and then you can apply it and monitor it. The whole goal here is not to keep rerunning the control over and over again. Some labs have always experienced controls that run outside a metric, so the first thing you do, you don't want to go just rerun the control. You want to go to your rules and see what the results actually mean. So that's the purpose of the metric. So I'll spend a little bit of time talking about the statistics. We won't go into this in depth. There's a lot in the literature. I'll give you some graphs to show you how you can apply them. I think that's the best way for a webinar like this. And as you go to the references, there's plenty of information that you can download these algorithms. So the sigma statistic, as shown here, sigma, you can see the equation. Its purpose is to describe the overall quality of the entire procedure. So again, the higher the value, the higher the quality of the test. So a sigma 6 is super, and a sigma 2 is not so good. The TEA, the total allowable error. Now this is an interesting metric because it must meet the medical decision criteria, and it makes up a combination of the random error plus any bias that's in the assay. And as I said earlier, for HIV, HBV, and HCV, for these viral load assays, the acceptable total allowed error is about a half log, so half log base 10. So that's total allowable error, and then random error as shown as standard deviation S, or in some cases SD, and then bias is systematic error from the true value. Oftentimes this is difficult to calculate, so what is the true value? And I'll give you some examples on how we use that here at SeraCare. Then you take the data and you plot it on a method decision chart, and it's a nice, easy graphical representation of the performance of your assay. And you plot on the Y the total allowable error, and on the X the imprecision. Okay. So this is a method decision chart, And as I said earlier, it's a graphical tool to evaluate the quality of these tests. And you should use it during your method validation and when you're taking on new assays to assess the acceptability of your performance of your assay on a sigma scale. So what we did is took some data. To give you an example, we took some CAP survey data on qPCR assays, and the bias was determined as the deviation from the mean from method A compared to 3 or 4 other methods. So method A is kind of the benchmark. And this is all from published data. And then from replica runs from this procedures, we're able to establish any random error as well. So here's a plot of the method decision chart. And again, we're plotting allowable inaccuracy or bias on the y-axis, going from 0 to 0.5, which for this particular application is what we've set the spec at, and the allowable imprecision or standard deviation on the x-axis. And you can see the sigma lines 2, 3, 4, 5, 6. Now, so we have 4 data sets here. We have Abbott Real-Time, the blue circle, Roche-Cobas assay, the pinkish circle, and the diamond in the dash being Siemens BDNA and a BDNA assay. So this is actually shows some, all these assays are performing very well. And you can make some comments that perhaps the Roche-Cobas system is the closest to the accurate values. But all in all, you see very good performance here between 3 and 4 sigma. So that's a good, quick evaluation of using these commercial tests in this application. So that's the method decision chart that you should consider right in the beginning. So then, after doing that mathematics and getting to your method decision chart, you need to assess your quality on a regular basis. So now we'll go back to how we use the ACCURUN controls for HIV-1, HBV, and HCV. And I'm going to show you some real examples using our in-house data. And again, the purpose is to monitor and show when an assay is out of control. And we'll apply the data to the Levi Jennings control charts, which I'm sure you've all seen. It's a very nice visual on following trends over time. And then we're going to apply the Westgard rules and Westgard rules in the OPS charts. And you can easily access these formulas and these equations on the Westgard homepage, which is in our references. So the products that we use for this data set are HIV-1, the ACCURUN 315. You can see the regulatory clearance. We actually have 4 different series at different concentrations that you can see on our website. HCV is ACCURUN 305, same thing. We have 4 different concentrations, and HBV is the ACCURUN 325 with 3 different series. And remember, we manufacture these controls with real virus in a commutable matrix to represent the patient sample. So the first thing you have to consider is the average levels for these controls. And statistical guidance always suggests running as many as you can. We like to recommend 20 replicates for any new control so you can establish a mean in the laboratory. So it's ideal to run 2 dupes over 10 runs. Now, if that's not feasible, and some of these NAT assays are very expensive, so it may not be feasible from a cost perspective, we're going to give you a solution where you can access other data produced by other customers for that same lot of ACCURUN control through our equality peer review database system. And you can use this access to this data to supplement your own data. So equality is a peer-reviewed database sponsored by Seracare, and you access it through Seracare's website, where your lab can post and review lot-specific results. And you can compare yours versus your lab over time, and you can compare your data versus other labs' data over time as well. And this allows you to, as the example shows here, so say you couldn't run 20 samples. But you could run 3 consecutive days in duplicate. You'd have n equals 6 for that specific control lot. And then you could go to equality, and you could look for that same lot number of control and grab some additional data there, some additional replicates. And say you grabbed another 12 or 14. Then you could do a one-way ANOVA test to show the data is similar, and if they are similar enough, then you can combine those data sets and save yourself some money and give you more surety for the actual average value for those controls. As we go through the results, I'll talk about what the actual control values were for each one of these qPCR assays. So a Levi-Jennings plot, as I said earlier, it really shows the results over time, and you can set your control limits in 2-3 standard deviations from the average run, and it gives you a nice quick visual to see when a procedure is out of control or trending out of control. And what we're going to show is actually normalized Levi-Jennings plots. So in this case, it's the observation minus the expected value. So the expected value is the average for that lot. And we show the graphs with plus and minus 2 standard deviations or 3 standard deviations. And so the y-axis is actually centered around 0 since these are normalized. And the next 3 charts are shown with data that were generated in our own laboratory, either here at the Milford site or at a Gaithersburg, Maryland site on the Roche Cobas system. And we've used the 20 measurements for a training set to get to the averages for each one of the controls. This chart is HBV, and it's the ACCURUN 325 run from January 2013 till almost early September. And you can see the blue diamonds are the training set and average to 0 on the y-axis. This is all log transform data, and I should say that you should always make sure that the units, either logs, international units per mil, or copies per mil, are always unified and do log transformations for these quantitative virology assays. And you can see the red line being the 2S and the orange line being the 3S. And as you can see with the HBV, there are a number of dots, or excuse me, squares that fall outside the 2S. So there is a little bit of imprecision with this particular assay. We'll come back to what that means here in a bit when we get to the OPS charts. So that's the HBV, Hepatitis B virus. Here's the HIV assay, and again, blue diamonds are the training set. The green circles the lot of ACCURUN 315, lot number 119772, actually run in our Gaithersburg facility. We actually moved that instrument to our Milford facility, and you can see we ran the same lot. Now this is an example. Say you wanted to move your instrument. Here's a good way to compare results from one physical laboratory to another physical laboratory, even within the same company. So again, you run the same control over time. And you can see we have just really 2 outliers out past 2 standard deviations. And you can see the HIV is very consistent over the course of time period here. And again, it's about, it's actually over a course of more than a year in this case with the HIV. Now moving to HCV, again, the same model. You can see the training set normalized around 0. And again, a very good assay, all the data points within 2 standard deviations. So that's a nice quick way to see how the controls are running in these assays. So now, how do we apply this data to an acceptance or a rejection rule for management within your QC of your laboratory? So in this case, we're going to recommend using using the OPS charts. And these OPS charts, first and foremost, have the same setup as the method decision chart. And what you do is you plot the observed inaccuracy, or the percent bias, on the y-axis, and the observed imprecision on the x-axis. In this case, a percent of standard deviation of total error. And then the operating lines represent the various rule sets to follow to identify when a process becomes out of control. If you go back to one of my earlier comments, so is that out of control from normal biological variation, or is that out of control due to assay drift or reagent lot change? Or is it a completely true assay and it's just normal variation, or the patient is undergoing some sort of therapeutic response? Now, all of this information is readily available, and highly recommend going to the westguard.com site to get these formulas and graphs. So for the OPS chart, now that we've got the Levi-Jennings data plotted, we want to see how the controls are running and what rules we should follow. So we're going to follow this data. And again, what the OPS chart we're showing, total error is going to be set at 100% of the medical tolerable variation. Again, back to my earlier comment of 0.5 log for HIV, HBV, and HCV on these viral quantitative assays. And we're setting the analytical quality assurance at 90%. So 90% of the processes out of control will be identified. And then we plot the assay bias versus standard deviation as a percent of total error. And then after you plot your results, you locate the nearest operating line just above that data point to establish the rule that you want to follow. So here's the 3 data sets on an OPS chart, and we're using n equals 4 for the rule coverage. And again, total allowable error is set at 100% of the 0.5 plus or minus log values, and with a 90% error detection rate. And you can see the lines are drawn as a maximum limit. That's a very poor quality assay. 1.2S rule all the way up to a 1.3.5S rule. We don't have the time to go through all these rules, but since these fall very close to the 1.2S rule, you can see the HIV, which is the squares, the HCV very close, pretty much just under the 1.2S line. And as we saw, a little bit more variability in the HBV assay, just to the right of the 2S, is that we would choose the 2S line for these particular assays as the rule to follow. Now, the 1.2S, since it's the most efficient rule from Westguard rules, this would mean that the probability of a false rejection, that rejecting a false rejection rate, is just 18%. So you can use that information when you see a control falling outside of the 2 standard deviation on your Levi-Jennings plots. So that's a quick synopsis of how to use the OPS charts. And again, I refer you to westguard.com for information about those. Now, if you do have drift in the assay and you see values n equals 4 outside of the 2S, as we just showed, then you have to do some troubleshooting. And here's a short list of ideas to consider if your control is outside the expected range. You could review the trends over time. Is there a trend versus high versus low versus the established mean? And of course, Levi-Jennings is a nice, easy way to do that. You want to confirm that the proper reagents, calibrators, and controls were used, and of course, stored the right way and handled properly. You want to make sure that your instruments are calibrated and up to date, and all the preventive maintenance records are up to date, and lab tech training is up to date, and everybody's used the instrument for all the procedures, have all the new training aspects to that particular protocol are followed. And I like to use the word and emphasize follow precisely. And then, of course, you can follow some other variables like lab environment to consider to help troubleshoot if, in fact, the assay is drifting. So this chart is a list of not only the ACCURUN controls that we used for this study, but all the other nucleic acid controls that are available from SeraCare today. So the top 3 I already spoke to. And then we actually have a multi-analyte control in HIV RNA, HCV RNA, HBV RNA. That's the ACCURUN 345. Again, the IVD CE and Health Canada mark. And we've got one series in 2 different kit sizes for this product. But we also have CTMG controls. We have HPV controls, both HPV DNA and HPV RNA, which is the most recent new product that we've launched. And we actually have a genetics control for factor 2, factor 5, and MTHFR. That's ACCURUN 611, also another new product. And this just over the last 6 months, we introduced actually a C. diff control, which is ACCURUN 501. And that's a control set with 4 unique Clostridium species that you can control for. So we have a nice long list of ACCURUN controls. And of course, you can get all this information by either visiting our trade show booth or the website. Now, I think one of the most important messages from this presentation is to remember how the daily run controls are central to any QC lab program. So you can use these run controls, these third-party controls, evaluating new kits, training new technicians, lab-to-lab comparisons, lot acceptance, inter-method comparisons, inter-site, and of course, and most importantly, troubleshooting when assays start to drift and fall out of specifications. And the data analysis documentation, everything I talked about with sigma metrics, you need a common control to be able to generate data that's associated with those metrics. We have a number of other products to consider. In addition to run controls, we have serology controls. We have performance panels that you can purchase, and linearity panels are very attractive for testing the linearity of your assays, but also the sensitivity of your assays. Qualification panels. We have a number of high titer isolates, the AccuType isolates. We have seroconversion panels and disease state materials that you can search our website for that will help you develop interesting new assays that you might be adding to the menu over time. And of course, we've got a good list of diluents to dilute your working samples. So in summary, we've talked about quantitative virology assays and how they require full process controls. We at SeraCare go to extremes to use stable viruses manufactured at defined levels to support your QC. From the metrics, total allowable error is, you need to know that, and the medically allowable error for HIV, HBV, and HCV is 0.5. The ACCURUN controls were developed and manufactured to be commutable, stable over long periods of time to allow you to compare results over a long period of time and allow you to monitor your assay processes, and are critical components to any of your QC management systems. And the metrics and the rules, provide a nice, convenient, easy way to use them to follow your processes. So here's the long list of references. Again, you can get them from SeraCare at our booth or at our website. And let me just, on behalf of everybody here at SeraCare, I want to say thank you for joining us at this seminar. I'll be at the booth to answer any questions that you might have. And I'd also like to introduce Megan McCutcheon, who's our Associate Product Manager. So there's Megan's email as well. So why don't I stop there and see if we have any questions.
Thank you, Dr. Garlick, for that informative presentation. We're so happy to have you part of the Cardinal Health Lab Exchange. Before we get started on question and answer session, I want to remind our audience how they can submit questions. They can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. So the green Q&A button is at the lower left of your presentation window, and we'll see if anybody has anything to ask. Okay, probably heading all over to the booth. I want to thank again our sponsor, SeraCare, for making today's educational webcast possible. Please make sure that you head over to the SeraCare virtual booth following this presentation, and you'll have the opportunity to engage in live chat with Dr. Garlick and your peers and have any other questions you have concerning assessing quality with ACCURUN controls for quantitative viral load assays. I also want to let our audience know that this presentation will be available for on-demand viewing through October of this year. And you'll receive an email from LabRoots alerting you to when this broadcast is available for replay. We invite you to forward that email to any of your colleagues who may have missed today's live event. Thank you for joining us. Hope to see you over in the SarahCare virtual booth. Thank you very much.